Today we will learn about mango anthracnose. It is very important disease of mango. High disease severity may result to heavy post-harvest losses. These are the various pathogens responsible for causing mango anthracnose. Colletotrichum gloeosporioides. Colletotrichum gloeosporioides were minor. Colletotrichum acutatum. The teleomorph stage of the pathogen Colletotrichum gloeosporioides is Glomerella cingulata. This is about the pathogen Colletotrichum gloeosporioides. It has haploid mycelium. The mycelium is septate, which are hyaline during initial stage and later on becomes slightly dark in color. The fungus produces acervuli as its asexual mode of reproduction. One or more conidia are formed from the apex of each conidiophores. Conidia are hyaline, but in mass they seem to be pinkish. The teleomorph stage of the pathogen, that is Glomerella cingulata, produces perithecia. Now we will see about occurrence of the disease mango anthracnose. It is known to occur in Brazil, France, Philippines, Indonesia, Trinidad, Peru, USA, Portugal, Hawaii. In India, it is common in UP, Punjab, West Bengal, and many other areas where mango is grown. Disease appears as blossom blight, leaf blight, wither tip, dieback, and on fruits. Now we will see what happens in blossom blight. The pathogen kills individual flower. The pathogen may produce minute black spots on inflorescence, resulting to drying and shading of flowers. The crop shows poor fruit set. Early infection of fruits result to premature fruit drop. On leaf, irregular black necrotic areas are observed which may collapse. Sometimes spots are of oval shape. Wither tip or dieback. Here, the wither tip or dieback appears at tip of young branches. Black dead areas are formed on affected twigs. On fruits, rounded brownish black lesions are observed as shown in the picture. The spots are slightly sunken and may have surface crack. During favorable condition, fruit lesions may extend up to pulp. Otherwise, they are generally observed up to peel. Epidemiology. Primary source of inoculum for the pathogen are diseased twigs, leaves falling on the ground. The fungus have the ability to survive on dead twigs. Conidia are produced on diseased flush, flowers and leaves. The production of conidia is favorable at temperature of 10 to 30 degrees Celsius and relative humidity of 95 to 97%. Humid and misty conditions are favorable for disease development. Now we will study about management. For management purpose, we have to keep in mind that plant should be in a good vigor, follow integrated nutrient management practices. Trees should be watered during summer season, prune and burn diseased twigs so as to reduce inoculum buildup of the pathogen. After pruning, spray with Bordeaux mixture 6 is to 6 is to 50. This should be the rate of lime, copper sulfate and water. Use copper fungicide. Follow hot water brushing. 15 to 20 seconds of hot water spray need to be done, followed by fruit brushing. The bioagents like Bacillus cereus, Pseudomonas fluorescens are used against this pathogen. The yeast Rhodotorula minuta is a strong antagonist to Colletotrichum gloeosporioides. Prabhakar et al. 2007 has reported significant efficacy of Trichoderma harzianum and Pseudomonas fluorescens. Spraying of fungicides should not be encouraged, as we know that they are not eco-friendly. However, if required, it is observed that zinab, copper fungicides, azoxystrobin, 75 mg active ingredient per liter, along with paraffin mineral oil of 0.2% is effective against the pathogen Colletotrichum gloeosporioides. Regarding the fungicides which we have mentioned in third line, zinab or copper fungicides, they are also found to be effective against the disease. Thank you.